Joyce said that today this session is more like an interactive session. Instead of we go over all the detailed protocols, I will just briefly introduce the different method, and we will share our experience with you. So if you guys have experienced some difficulties, and if you have any troubleshoot, and if you have your own experience, we can share during our discussion sessions. So when using Agrobacterium for plant transformation, the first step is introducing your new construct into Agrobacterium strains. There are 3 different methods that are commonly used for Agrobacterium, including electroporation, freeze and thaw method, and triparental matings. So for me, it's electroporation is the way to go because it's very simple and highly efficient. Therefore, I have used electroporation maybe 99% of the time. It's good to have it. If you have the equipment, it's really simple, but it also has some downside, including the equipment. You have to purchase the micropulsar or gene pulsar, or second, the consumables such as electroporated cuvettes. They might be expensive for some laboratories. So in that cases, freeze and thaw method or triparental mating method are more appropriate. So we provide a link to different protocols. If you actually Google search, there are maybe, if I exaggerate a little bit, there are more than 100 different protocols. In principle, they are very, very similar. You need to grow Agrobacterium to, let's say, log phase or early stage stationary phase, and then you harvest the cells and chill them, and you need to wash away all the electrolytes, right? And after that, you have to concentrate them. Usually, the final concentration will be like a 5 times 10 to the 8 cells per electroporation. And then you need to do the outgrowth a little bit. So yeah, this one shows a micropulsar from BioRed. And as you, let me have the pointer. What is that? Connections. Sorry, I couldn't find pointer here. So this is the electroporator, and there's a chamber here, down here. Yeah. And on the right side, these are 2 millimeter electrocuvettes. They cost about $2 to $3 per cuvette. So for some labs, it could be too expensive. And you have to chill them on ice and add the DNA, mix the DNA with the cells, and add into the cuvette and put into the chamber, and press the pulse button here. And usually it's a high voltage and no current or 0 current. And the amount of DNA and the competent cell and then cell density, they all affect the outcome of transformation. But this method is highly efficient. So usually you get many colonies from the plate. Next slide, please. Yeah, this is the typical outcome on a YP or LB or MGL medium plate after 2 or 3 days of incubation. So make sure that there are sufficient number of colonies there, meaning that your transmission efficiency was good enough. And when you grow them, you are not supposed to pick the tiny small colonies. They could be false positives, or maybe the plasmid are rearranged. So always put well isolated single colonies, the large colonies. And then it's important for you to check the plasmid DNA integrity within the Agrobacterium, which we will discuss more in the quality check session. Next slide, please. So here are additional links to the freeze thaw method and then competent cell preparation protocol. Freeze thaw method, it's relatively less efficient than electroporation. However, you can still introduce the your construct into the Agrobacterium. So you prepare the competent cell, and it goes to freeze and thaw, meaning that once you add the DNA, after thawing the competent cell, you have to freeze using liquid nitrogen or dry ice, and then you have to thaw them again, either at room temperature or 37-degree water baths. And then you, after outgrowth, so you spread them and you grow them. So this requires more cells, and it actually takes a little bit longer. And tree-parental mating method is another method which requires 2 additional E. coli strains. One is the helper strain, which carries the conjugal transport machinery. And then your target plasmid that you want to transfer to the agrobacterium, you need to mate them along with your target agrobacterium strain. So it's like a 2 E. coli strain and your target agrobacterium strain. And this takes longer than the others. It takes about a week to introduce. However, this method can handle large plasmid DNA, but there's just some additional requirements, such as your construct must have the origin of T to utilize this method. So I believe we went over these 3 methods: electroporation, freeze and thaw, and tree-parental matings. So now I would take any questions if you guys have.
Actually, Consub, we're going to take any kind of inputs and tips. You missed the early part from the steering committee, from the panelists,
okay, sure.
and then we'll open it for questions.
Okay.
So for instance, I'm just going to go back to the electroporation to give some tips on what we do for an agrobacterium strain called AGL1. So my lab primarily uses AGL1. It's our favorite strain. And what we do is that we take an aliquot of 25 microliters of our, as Consub mentioned, we have a glycerol stock. We make the glycerol stock small enough that we're not repeatedly freezing and thawing, because that can affect the growth of the agrobacterium. So we do ours in smaller aliquot. So when we have our plasmid DNA, we've done the nanopore, we know the concentration. We then take an aliquot, 25 microliters of AGL1. We add 1.5 microliters of the plasmid DNA, and then we do the electroporation. And after the electroporation, there's a recovery period, because the electroporation works by opening up pores in the bacterium for the plasmid DNA to go in. But then you need to give those cells time to recover so that those pores close. So what we do is we put them in a medium called SOC, and we let them recover for 1.5, like 1.5 to 2 hours recovery on a slow shaking. Then when we aliquot them onto our plates with the appropriate antibiotics, we actually do like 1 microliter from that aliquot and 5 microliters. The reason is if the electroporation worked really well, then we would get like a lot of colonies and wouldn't be able to pick out a really nice single colony like Consub showed. So we do a 1 microliter and a 5 microliter aliquot. And then we only pick, as Consub mentioned, we only pick those nice, what we have by experience know are the good size. And Consub show those really small colonies. And you have to be careful, because even sometimes when you do an electroporation, every colony will be that small. It's not just a mixture, and that indicates there's a problem. So for instance, we got an aliquot of plasmid DNA from a collaborator once, and we grew up the cells after electroporation, and they grew much slower. So instead of taking 2 days, it took 3 or longer, and the colonies were like pinpoints, very small, and we knew there was a problem. We had seen that before with other aliquots from a collaborator. Well, what happened was when they did the sequencing of the plasmid that they gave us, they found that a point mutation happened in the selectable marker in the bacterial and the backbone of that vector. So the kanamycin selection, which is what we were using, it wasn't expressing high enough for the colonies to survive. So they had to go back to their original stock of vector and remake that for it. So just some tips of what to look for, like how we do that with AGL1, that would be specific for different agrobacterium strains, and then just some things to look out for. Any other comments from the panel before we go ahead and take questions?
I would like to add that Consub showed equipment, and then there are different types of equipment for electroporation. So it's very important for user to understand what that is, because some of these equipment are capable of fine tuning and adjustment. And if it is a multi-user equipment, then you have to make sure you are using these settings that are meant for Agrobacterium strain electroporation. So that is one point. So know your equipment, and then know your Agrobacterium strain. And then second thing is, if you are not used to this, it's very important for you to run the control. And then control would mean that you have, the competent cell, you have an empty vector. And that can also help you understand if you are finding different size of colonies, or colonies that are not capable of utilizing or taking care of the antibiotics, or growth in the presence of antibiotic, or the effect of the gene you are putting in on the agrobacterium growth. And so running those controls, at least when you're changing the competent cell or the new plasmid vector, it's at least important to run it at least once if you cannot do every time. In our lab, we do it every time when we are doing it. So that will be all from my side.
So I have two questions from the chat before we move on. So the first is from Nadia. For triparental mating, how large is large DNA fragments?
That's a good question. I have not tested it, but it's over 100 kb has been used.
Okay, great, thanks.
Mm-hmm.
And then this is from Quazim. Sorry if I mispronounced that. Which method of plasmid transformation works best, chemical or electric shock?
In terms of efficiency, electroporation is very efficient. And it does not require any requirements, such as triparent mating. You need to have the origin of a transfer. Not every construct you can do that. That adds additional size to the construct. In addition to the left border, right border, and original replication for agro, you need to have the origin of a transfer if you want to utilize the triparent mating.
Okay, and then I have another question here in the chat from Masi. So kind of a follow-up question to what we were talking about is the method you use, talking about electroporation, freeze thaw, or triparental, is that based on the plasmid you're using or the Agrobacterium strain?
I think that's another great question. For triparental mating, I only know from this literature because the conjugal transfer machinery is provided by E. coli, helper E. coli strain. I think any Agrobacterium strain can be used as a recipient.
Okay, well, thank you for those questions. There were a few other questions in the chat. So I opened up the chat. People should be able to reply to other people's questions, and then also you can use the Q&A option too as well. So I'm moderating both of those. So for our next speaker, we have Vina from the Donald Danforth Plant Science Center.
Hi, everybody, and I'm going to talk about long-term storage of Agrobacterium strains. There are a couple of different ways you could prepare the glycerol stocks of the Agrobacterium strain. This step you generally do either you are trying to make a competent cells with the Agrobacterium strain you have in-house or you obtained, or the reason you are going to make these stocks of the Agrobacterium strain if you have delivered a plasmid of your interest into the Agrobacterium, and then you would like to store it so that you could use it in future. You can make either glycerol stocks or DMSO stocks. These two stocks for in order to keep these stocks for long-term storage, you need a -80. And these are the commonly used practices in the lab. And these are also applicable to E. coli strains. And you could also do the stab cultures where you are using the agar gel and then stabbing the culture with the stock or the Agrobacterium strain you have in hand and want to make a stock, and then store it at 4 degree or room temperature. The longevity of the stab culture is shorter compared to -80. And then some people have claimed like they can recover these strains 40 years or 50 years from the storage if they needed to from glycerol stock or DMSO stock, but stab cultures definitely have a shorter lifespan, and then they needed to be periodically maintained. The text in the blue in this slide shows some of the protocol we have obtained from the labs which follow them routinely. Please note that if you're picking a protocol, follow that what is written, and unless you understand why a particular step is being done. So it's very important to understand what each step means and why it is being done. And if you don't understand it, follow the way it has been described. Next slide, please. So when you are making these glycerol stocks, what are the things you need to keep in mind? The purpose you are doing it is for long-term usage. So you have to make sure you are using the correct strain containing the correct plasmid you intended to put in. It's better to make multiple tubes or in small aliquots. This is to avoid freeze and thaw. There are some people have tested, so freeze and thaw of the glycerol stock. It can survive or last for some time, multiple freeze and thaw cycle, but it is very non-reproducible kind of a thing. If some problem comes, then it will be hard to resolve. So it's better to keep it in multiple stocks, in small aliquots. The timing to make this glycerol stock is very important. You do not want to make the glycerol stock, which is a couple of days old. It's very important to make it an aliquot and the glycerol stock very early. For example, overnight grown culture. You should not be waiting for too long to make the glycerol stocks, such as storing it in 4 degrees or room temperature for a long period of time and then choose that, okay, now it's time to make the glycerol stock. If possible, it is good to try to grow to an OD as indicated or defined in the protocol if that requires. A very important thing, last but not least, is proper labeling and record keeping. The goal you are doing this is to use for long term. And if you don't label it well, then you lose the track and trace aspect of those things. And then you would not know what you are handling in future, or your lab mates would not be able to follow it if they are trying to follow later. Next slide, please. So are we at pause here? Okay.
Okay. So are you done, Veena? Are you done now?
Yeah, I'm done.
Okay, great. Okay, so I have a couple comments to add. Thank you, Veena. That was really a good overview of kind of the idea of follow the protocol. If this is the protocol you were given, make sure you follow that. And preparing competent cells is a really time commitment. It's we've got to make sure that you're doing everything properly because you're spending a lot of time. Just a couple of tips. One thing that we do is that after we make a batch of competent cells is we test them on the different antibiotics that we use for selection, just to make sure that we're getting either no growth. If we see some growth, then we know we might have a contaminant. If you see some growth on the antibiotics, then that might indicate that we have a contaminant of some sort in our competent cells. But we also, before you start using them, it's always a good idea to test them on the different antibiotics that you're using for selection. Another thing is that if you do it properly, I remember we had a workshop and I think it was Steve Strauss who talked about taking agrobacterium out that had been stored for 40 years. Was it like 40 years? I think it was from the 80s. And the agrobacterium continued to grow. So if their competent cells are prepared properly and also, stored properly, then they can have a good long life. And we're always very careful with our glycerol stocks, as I mentioned earlier. So say if we're using 25 microliters for our electroporations, you don't want to do like a 200 aliquot of your electroporation of your competent cells, because you don't want to be freezing and thawing so much. And we always put them on ice. We keep them on ice. We're being very careful about how we're treating them, because they're, we want to make sure that we have good batch of cells, and also that they're going to work well for electroporation. All right, that's all I have to say. Are there any other comments from other panelists before we take questions?
Okay, if there's no other comments, then there's some questions in the chat. So the first question comes from Beth. Can you recommend a free database for tracking agro stocks? We have been managing with spreadsheets, but a database would really make things much better. Would the free version of Airtable be sufficient? I don't know, Veena, do you want to talk about your process?
Yeah, so I do want to say this database, so there is no standard practice, or we can point to this is the Excel sheet or this is the database. You can go to these track and traces for your lab or your use and your lab purpose. And so more defined or more information you can put in, it's better. I believe at the end or somewhere at the end of this slide deck, you will find some example. However, whatever you do, best way to capture all the information which is needed. So capture it for others to use, not for you to know. Because you may know a lot of information already, so you can skip, but think about when you are entering these information for somebody who don't know. So that's how I go about, but Excel sheet, paper sheet, anything, as long as you capture well and it's accessible, is a good system to follow. If nothing else you could do.
I like that, for other people to use, not for you to know, because there's I think a big difference there. So another question was, how many times would you typically use one glycerol stock from the -80 before switching to another aliquot of the same glycerol stock? I don't have a problem with the agro growing back, but after 2 to 3 uses, it seems to decrease transformation efficiency.
So if Veena wants to go ahead, and then I'll go ahead.
If I have a choice, I would not freeze thaw. So that is how we practice. That's why we make multiple stocks, and so that we are grabbing one tube at a time and throwing it away. And then you will know, like, how if you freeze and thaw a glycerol stock, it depends on how well you are doing it. Because if you are taking the glycerol stock, putting it in dry ice, not letting it thaw, you could use it, and people have used it for long. But if you take it out, leave it on laminar flow hood, and then go out for coffee or long, doing other thing and then come back, that could be a problem. And then it's very situation specific, so it's hard to say. So shoot for not to thaw, to make life easy and less complicated.
Yeah, and I would like to say cold, cold, cold. Just if you don't have the space in your -80 and you're using a lot of different agrobacterium strains, and you can't make little aliquots, 25 microliter aliquots. When I was doing a long time ago, when I did my graduate work, I didn't freeze or thaw my aliquots more than once. I would do it in like a 50 microliter aliquot. I'd take that tube right out of the -80 and put it on ice, and then use it right as it thawed, and I'd get my aliquot and then put that right back into the -80. So only one thaw for mine, and then keeping things cold.
Yeah, I keep use the same glycerol stock for a longer time, but you should not let it thaw. You prepare everything in the laminar hood and then just take out the glycerol stock, one glycerol stock, and put on ice briefly. And then you immediately inoculate, just to touch the top, scrape a little bit from the top. And before it thaw, you put it back to the deep freezer again. And this way, you know, as long as it's not thawed, it can be used multiple times.
Yeah, and my experience is similar to that. You know, using multiple multiple uses of the same glycerol stock, but when we take it out of the -80, you know, we would put it in dry ice, and keep it in the dry ice while you're doing the scrape, if possible. So it never really comes out into the atmosphere to thaw at all. And, you know, it might have been a little bit of overkill, but, you know, we didn't have any problems doing it that way. So.
And I have one more question in the chat. Is DMSO storage better than glycerol?
I would like to say it is one of the method. And so I do, and other thing I want to point out, I did my postdoc with Stan Galvin. So we made 4 glycerol stocks, 4 stocks, 2 from for the glycerols, and then 2 for the DMSO. And then we we were keeping that in 2 different -80s to prevent, because the effort you put in to make these glycerol stock or stocks of the agrobacterium, it's very, you know, tedious, time consuming. And once you have it, you want to keep it safe. You don't want to let it go or lose just because equipment failure, or you did not aliquot it well, or things like that. So I would say that is one of the method. That's all.
Okay, great. Thank you, Veena. Okay, and then there was a question about contamination, but I think we're going to talk about that in the upcoming quality control section. So Veena, I'll turn it back to you to talk about quality control.
Okay, is slide moving? Okay. Yeah, so previous slide, yeah. Okay. So quality control, anything we do in plant transformation, every step you can think of, I would say QC, QC, and QC, because it's a long process to generate a transgenic plant. Any failure or any misstep can impact your long-term goal, and then loss of resources and things like that. So it's better to check this early on in every step you could and whenever possible. And so QC confirm plasmid. Here, what I mean is when you receive a plasmid you obtain from a company or vendor or friend or a next door lab, it's very important to start with QCing that plasmid, because that's what you are using to create your recombinant binary vector for the transformation process. Once you add your elements or traits or the sequences you wanted to put it into that plasmid, you QC again. And when you're QCing, traditionally, when sequencing was very costly, you would QC just the region and restriction digestion of that plasmid to figure out if it is a quality or like everything went right or not. Nowadays, sequencing is cheap, so I would highly advise sequence the whole plasmid. That would allow you to sequence the vector backbone as well. And that can help reduce a lot of problem downstream. And so you got the plasmid, you QC'd, you put your sequences in, and then you QC. The first step you are doing is putting that plasmid in the E. coli. So you isolate the plasmid and QC that. Once you QC confirm, take that plasmid, introduce into the agrobacterium, take the plasmid out, either put it back in the E. coli restriction digestion sequence, or whatever way you would like to QC, do that, or else directly isolate the plasmid from the agro. In order to do that successfully, you have to obtain large volume of agrobacterium to isolate the plasmid and send it for sequencing. When you're doing that, keep in mind, nowadays we have ternary plasmid systems. So in order to sequence that, you may have to put back your isolate the plasmid from agrobacterium, put it back in the E. coli, select those E. coli for a specific plasmid type you have put in, and then send it for sequencing. So all these are very important. Any rearrangement in any of these vectors could cause problem downstream, and you may not get the intended outcome you are looking for the transformation experiments. So once you do sequence, please make sure you're updating the map, because nowadays many times I see people found these mutations, those information stays in the notebook and maps are not updated, and these propagated for the future user also. So it's very important to update those maps. It is part of the track and trace strategy for you and for future user. Watch out for any point mutations. Sometimes they are very forgiving where they are, but sometimes they are not. For example, Joyce Winnick mentioned there is a point mutation. It causes colonies to grow very slowly. It can impact again how well your agro is growing. And if it is stressed, it would not be happy source for transforming the plant cell. And so it's very important to check and log. And I already mentioned that, when you're doing the conventional binary vector where there is one plasmid involved, you could QC either isolating the plasmid from the agrobacterium, putting it back in the E. coli, or directly isolating the plasmid from the agrobacterium. But if it is ternary vector system, you may have to put the plasmid back in the E. coli and then QC, unless you have very strong sequencing platform in place, and then analysis software, which can directly analyze and then assemble the plasmid for the sequence you're looking for. Do and don'ts for this: ensure you're working with the agrobacterium type plasmids and the plasmid sequences you are intending to use. If you are choosing to use the AGL1, the method you are following, it involves AGL1 and a specific plasmid. In order to be successful, follow that. Don't make changes unless you know what you're trying to do. And once you get used to, you can expand your horizons and then follow and optimize as needed. And if there are errors and all those things, fix early and update map. I keep on saying this, but it's very important because that's where a lot of mistakes happen and propagates for future. Next slide. This is very nice example, and this is from our lab. And we were working on the plasmid. So first, if you are not routinely transforming plants in your lab, and then you occasionally do this, and then you hear that, oh, I did PCR QC my agrobacterium strain and looking for PCR confirmation for the selectable marker. And then I found the colony which has positive. So let's go ahead and move it forward. And so the left side panel, the first one, standard PCR of agro colonies, is the example of that. Here you can see there were 6 colonies, positive plasmid, positive control, then AGL by itself, no template control. So you see every one of those showing the positive fragment size. And then you did the full plasmid sequencing. And what you find there is there are 2 peaks. There is, most abundant plasmid in there, but there is a rearranged plasmid up there. And then when you did the restriction digestion of the plasmid isolated from the agrobacterium, you see multiple fragments. And then this is all coming from the same plasmid. And then what was happening in this, the lower profile, the lower peak you see in the middle image, that is what was intended plasmid right there by the researcher. And then it shifts the balance of the plasmid within the agrobacterium. It was predominantly predominant or dominant form of the plasmid was larger size of the plasmid. And so if you use this plasmid, did not QC, then you will end up finding the transgenic event that may have the rearranged tDNA in it, or maybe you will get less transformation, or it will be hard for you to QC the outcome. And so that's why it is very important to run the controls, and also very important, if you see some off observation, it's very important for you to follow so that you don't make a mistake or don't waste your time moving forward, because right now it is just the step of making the plasmid and then not spending a couple of months or year to generate the transgenic plant. So this was very nice example. So what we ended up doing is start the experiment right there and went back to make the plasmid again. So it was really neat example to follow. Can I have a next slide? Oh, okay. So we can stop in the previous slide and then we can ask Joyce, you would like to have open for Q&A here?
Yep. So if there's any other comments again from the panelists, and I just wanted to remind people that, you know, we're giving you an overview here, kind of leading you to where information is. We have the survey. There's a link in the chat. We'll talk about it later to go into more detail on each of these topics, because as I look through the chat and through the Q&As, there's some great questions there. We won't be able to get to all of them, but keep an eye, do that survey, and then we will have separate little interactive sessions on each of those topics. And also, if you want more information, background on Agrobacterium, go to the PlantGENE website. We have recordings from an Agrobacterium workshop that we did. And also we have other resources on the site where other workshops that we've done. So you'll get more of the background on Agrobacterium that we're not doing today, because today's more like you're in someone's lab and you're getting this practical advice. But please, I encourage you to go to the PlantGENE website and look at those workshops. Okay, any other comments about Vina's topic? If not, we can take questions. But if any
Yeah, I can add one comment.
Okay.
So I have used a different CRISPR-Cas9 system in agro, and some codon optimized Cas9 show the toxicity within Agrobacterium. So that means when you introduce your construct, it's not because of the toxicity, some toxicity, maybe all the colonies look very small. Even if all the antibiotic resistance gene is okay, no problem. It could be the toxicity of the gene element you added other than your selection marker genes. In that case, you may be able to find a different version of a CRISPR-Cas9 without toxicity. However, if you cannot avoid it, there's a way you can still culture and do the transformation successfully. So for those cases, you still have to do the QC, even if it's tiny colonies. You need to grow and make sure that your construct is intact within those small colonies. So it's more important then. So that's what I want to add here.
I would like to add one more quick thing. What Concept mentioned, that QC, it means, in my mind, it means, it's a stable version of the plasmid in the Agrobacterium. So please note that if there are, it is rearranging, it's not stable. So you need to find consistency when you're sequencing that it is the same plasmid stays. And then either also you can grow it in lower temperature, and then think there are some tricks you could follow. Thank you.
Okay, questions, Amy?
Thanks. Yes, so we have 2 questions in the chat. Vina, could you please elaborate on the different peaks you get after transformation? So I believe that was the slide.
Yeah, right here. And so thank you, Riaz, for this question. So you see there are 2 peaks. The plasmid, the full length right plasmid was around 12 kb in size. That was expected. And then what we noticed, this is an example of RNAi vector that was we were trying to handle and then do the transformation. And then you see that there is a population of the plasmid which is the sequence was assembled in silico that was showing 14 kb in size. And so these are based on the full plasmid sequencing. And so these sequences are fed on the software, and then these are assembled in silico, and that's what it shows and demonstrates to population. But generally what happens is if things are right, you will see only one peak, which is of the right size. And so in our lab, we try to sequence QC or QC our Agrobacterium strain and plasmid in multiple ways, depending upon what we are doing. And so I hope it answers your question. And then if not, Riaz, please feel free to send me a direct message and I will be happy to follow up on that.
And then
Thanks.
this question goes back to, we were talking about plasmid size and electroporation. So someone wrote, we have tried to transfer the CRISPR-Cas9 cascade. It's about 14 kb in size. Which method, is best suitable?
Any method. I've been using
Okay.
electroporation for up to 35 kb, and it works really well.
Okay, great. All right, and then I had answered both of these questions, but I just want to open this up to the panelists too. We had 2 questions about what to do if there's contamination. So if you have plasmid contamination, or if you have 2 strains of Agrobacterium. So I think both of those you would need to restart your experiment. But I'd be interested to hear from the panelists.
I would like to say here that if you have a agrobacterium contamination and like you could follow standard microbiology practices, however, you have to also figure out the strain is what it is. And then you may have to deploy other methods to confirm. So in those cases, I stop the work, like that's my guidance to the lab people, is stop that and obtain the strain from the source who know, from the place where they know what they are doing, and then they have the high quality strain they can confirm and go back to the source again, rather than trying to sort it without knowing how to sort it.
Great, thank you, Veena. Okay, so with that, we will move on to our next section, which will be covered by Bill and Joyce. So Bill, we can start with you.
Okay, so thanks, Amy. So there are various protocols for growing agrobacterium and different media. Depending on the agro strain you use and the plasmids that you have in the agro, there are going to be different combinations of antibiotics that you use. So if you can go to the next slide. So I just wanted to stop here for a second. So, we developed this LBA 4404 agro strain as an oxytrophic mutant. So it's a thymidine minus mutant, and that really helps to control agrobacterium after transformation. Because sometimes antibiotics aren't strong enough to rapidly eliminate growth of the agrobacterium. And with some species and with some explants, this can be even worse. It can be a real problem. And so this worked really well for us. But LBA 4404 is not as virulent as, for example, AGL1. So we built a virulence plasmid that we use in LBA 4404, and it works just great. And one of the other reasons that we like this agro strain is that it works really well for growing on solid media. And we found that, for the different labs in our company, for new people coming in, that this was a really simple method to use. So that's a little background about this strain and why we use the solid cultures. But I also wanted to just point out, details matter so much. So if you looked at our leaf transformation paper in May's last year, we lay out all the details for doing this culturing on solid media. But a lot of people get our strains and they say, oh, we don't use solid media, we're going to use liquid. And if you use the same media in liquid, the agro strain behaves very differently, and it can cause problems. And so just again, details really matter. So this is a very simple example of pointing out the details of how we streak out agrobacterium on our solid plates to prepare it for transformation. So next slide. So when we bring our agro out of the glycerol stock, we just want to streak it out, and this is a real standard microbiology protocol. You want to streak it out in a manner that's going to dilute it to the place where you can get single colonies. So we take our loop with the glycerol cells and we streak it on a portion of the plate as shown in the red there on 1. And then we take a new sterile loop and then we streak across some of those previous streaks over into a fresh section of the plate. And then we take a third sterile loop and we drag it through the last streaks and bring it down into the fresh media in the remainder of the plate. And if you go to the next slide. And so this is the type of result that you get. You start off with, just a confluent streak of Agrobacterium. and that's really not what you want in this case. You want to be able to go in and you want to be able to pick out single colonies, then to go to the next step, which is to grow your agro to log phase so that you can use it for transformation. And we call these the working plate. So we pick that single plate with a sterile loop and we would streak it across the plate. And you can, and what we're looking for here now is not necessarily single colonies because we already picked our single colony. We're just looking for enough growth overnight that we can go in and we can pick up some of this agrobacterium and put it in the liquid infection media for doing our transformations. So this is how we make a working plate for immature embryo transformation. And then we would go in, I'm sorry, you can go to the next slide because this shows the result of that working plate. And then we would go in and we would just streak out, for example, down at the bottom. I typically like to streak out some areas where it's not super dense, super heavy, because typically in those areas where the agrobacterium is really crowded, the growth might have already started to slow down. So I would pick an area that was less dense, but you need enough agro that when you put it in your liquid for infection, that you're going to be able to get it up to the proper OD. And so now we can go to the next slide. And so that was the example for immature embryos. For leaf transformation, we use a much larger volume of agro suspension. Instead of 1.5 mils for immature embryos, we're now using 100 mils for leaf transformation. And so we just want to cover the plate with as uniform a confluent covering of agrobacterium as we can. So we just start streaking the plate in the upper left there, and then using the same loop, we would turn the plate, 20 degrees, 25 degrees, and streak again, and then keep turning it and keep streaking it. And I like to do this for a couple minutes. And what you want to do is you want to get as confluent a growth as possible. And I would almost say that this might actually be a little too dense. So that's something that you want to work on. You kind of empirically want to adjust that. I like confluent growth that where the aggro isn't too thick, because I think under those conditions, the cells are still growing rapidly, and we get better transformation results. So you don't want this to be too thick. So that can be an issue. So that is all I wanted to say. So,
Hey, Bill, like, what you would advise, like L loops kind of thing, the spreader for that method you just mentioned the last, that can people use the spreader?
so yeah, sure. I mean, we typically just use a sterile loop when we do that, just because it's something easy that we have right in our transformation hoods. But you could use a rotating plate and a spreader. That's a great idea. I mean, we do that in our E. coli lab, but we don't do it with our agro. But you're right. That could work well.
Joyce, can I add more comment if you have time?
Sure. We're going to go to
Okay.
liquid culture next. If you want, let's wait till
Okay.
the end. Let's wait till
Okay,
the
yeah.
end. That'd be great. Amy, could you go to the next slide? This one isn't mine. Okay, so thank you. So as Bill indicated, that follow the protocol you're given for the preparation. You know, Bill talked about solid plate and how they do their work, but there are protocols, and the kind of the method that we follow in our lab is where you go from a plate of colonies to an overnight liquid culture. And you see here in the image on the left, I show a plate that has some colonies on there. And what we use, you can use sterile toothpicks. We've started using sterile pipette tips for picking a colony and putting that into an overnight liquid. And you'll see here on the right, this is a bacterial medium that has a toothpick in it that had colony on it. And then we put that into a 28°C incubator. Now, probably a lot of you know that you don't want to go over 28°C when you're growing your agrobacterium. That causes instability. I mean, basically the agrobacterium is not happy, and it starts kicking out non-essential type of components that'll affect its growth. So you want to stay at 28°C. We do shaking overnight because you want to have the aeration there. Amy, if you can go to the next slide, please. And then overnight, this is what our culture looks like. Now, we always read the OD of our cultures because we want to be consistent with each of our experiments. I know I've interacted with people who say, we don't read the OD. I can, if that's what you want to do, that's fine. But for us, we feel that we really need to do the OD because we're putting so much effort into the downstream transformation. It doesn't take that long to read the OD. If it overgrows, if it overshoots, oops, I'm not done. Sorry. If it overshoots that OD, you can dilute it back, let it grow for a little bit more time, and then and then look for that OD that you want. And the OD will be in the protocol that you're following. Now, I saw in the questions and answers that somebody said that sometimes, so when they do their bacterial cultures on the plate, they grow well, but when they grow into a liquid medium, they don't grow well. And I can share an experience we had with LBA 4404. When we grow the medium on the bacteria on an LB medium, the bacteria LBA 4404 grew really well. But when we went into an LB-based liquid medium, the agrobacterium was chunky. It didn't have this nice, as you can see here, well-dispersed bacterial cells. So what we ended up having to do is we grow the, we would grow the LBA 4404 on a solid plate of LB, but then when we went to liquid, we would use a medium called YEP, and then we would get this much nicer, well-dispersed growth. So that answers one of the questions in the chat, or question and answers. Amy, can you go to the next slide? So after we've read our OD, we then spin down that whole culture, and you'll see here on the left, we've got these really nice looking pellets. We take off that bacterial medium. You want to take off the bacterial medium because it's high salts, and the plant material are going to, not be happy. They're going to react badly to that high salts medium. And then we resuspend that pellet, and you'll see here on the right, in a liquid plant-based medium. It's basically an Mershigi Skug-based medium that then we use for our transformations, okay? So we want to, and we're going to switch into the antibiotics next. So that's the next section. So those are just some tips that for growing it in liquid, from a plate to liquid. And so you've seen two different methods, the way Bill presented it, and then the way I presented it. So, okay, now we can take some comments before we go on to the next section, antibiotics, comments from the panelists. Vina, if you wanted to share what you were going to say.
Yeah, one of the thing is you heard Bill talked about, and there was a mention of the temperature, the equipment temperature. And so please make sure when you're dealing with Agrobacterium, don't mistake it to the 37-degree E. coli, because there is a potential for losing the TI plasmid. And so if a equipment says it's 28 degrees, check by other means also if it is an older equipment or it's not calibrated. So please, it's very important. And then also you have heard, liquid method, you heard Bill's method, and then in some labs, they exclusively recommend to start with the single colony. And so it's very important for you to read the protocol, what you're following, and follow the guidance until you become pro on that method before you go for changing, or try to understand why a particular method is done, because mixing methods can lead to difficult outcome, which you will waste time on. So it's very important, follow the method, and details matter, as Bill said.
One thing that reminded me when Vina talked about temperature, what we do is we don't rely on the temperature that's reading out on the outside of an incubator, because that's not always accurate. We'll put a flask with a thermometer in there to make sure that we're calibrating our incubators to the actual temperature inside the incubator versus what's on the outside on the controls, which might have to be higher or lower to give you the exact temperature that you want. Okay, a couple of questions, Amy?
Yes. So first question is from Lena. What is the goal in using 3 different sterile loops when spreading bacteria on solid media instead of using 1 sterile loop? Is it that you're likely decreasing the amount of bacteria with each successive loop?
Yeah, so using 3 different sterile loops dilutes the agrobacterium each time that you're doing a streak. So you streak once with 1 loop, and then you take a fresh loop and you just pick up a little bacteria from the first streaks and you streak in a fresh area, and then a 3rd loop you do it again. And so each time you're diluting the bacteria, because you want to get down to those single colonies.
Right, that makes sense. Another question from Sudeshi. Do you add the antibiotic to the broth at the same time you start the culture? Or is it okay to make a stock broth containing antibiotic and store it in the fridge to be used when needed? I think that one's for Joyce because it's about liquid culture.
Oh, okay. Yeah, I think it's always best to add it right before you grow it, because if you're adding it and then storing it, your antibiotics are kept in the freezer. We keep ours in the freezer. And even in a freezer at -20, I can tell you all these little tips and things we learned throughout our careers is that they're not completely stable even in a -20 freezer. We found when I was a graduate student that antibiotics stored in the freezer for longer than 6 months weren't as effective before that time. Anyway, I think it's always best practice to add, especially your antibiotics, right before. and not put it in. They're stored in a freezer for a reason. When you get antibiotics in a solution, they become more unstable. So over time, they're not going to be as effective. So best practice I would recommend is to do it right before.
So a couple other comments there. Some antibiotics like tetracycline are light sensitive. And so you have to keep that, once you put the tetracycline in solution, it has to be kept in the dark. And when you mix up solid media with antibiotics, you cannot add the antibiotics before autoclaving because a lot of these chemicals are not very stable in the autoclave. So you have to autoclave your base media, let it cool down just before it's starting to solidify, and then mix in some of these chemicals like antibiotics that are more sensitive.
Great, thank you. So this is another question for Joyce. You mentioned that you had some clotting of your agrobacterium. Gopika is having the same issue, and they asked, why is this kind of growth occurring?
So it indicates that, not that I want to give them a characteristic, the agrobacterium is not happy. So as I mentioned, what we found was we would, like for LBA 4404, we would go from a solid plate of LB medium, but then when we went into a liquid medium that was an LB based, we saw that stringiness, and we switched to YEP liquid medium. So solid LB, liquid YEP. So I would recommend that they try different liquid bacterial medium that you'll find in the literature for growing agrobacterium to make sure that they get a good dispersed growth like that. So
one thing is agrobacterium tend to form biofilm. So it is, whenever they do clump together, those bacteria may not be available for infection. But there is, you can read about this in literature and publication from Stan Galvin and all those things to understand more. But, and then varying the media can change the outcome, but there is a lot more to it. And yeah.
if there aren't any more comments, we should move on because we're past the hour here.
Yes, so this is last question, and it's two questions from two people that were related. So how sensitive are agrobacterium to centrifugation / sedimentation? What is the speed limit? And once you centrifuge, how long can it be kept without the media?
We've never tested that. We just follow the protocol that we were given, and we immediately resuspend and then use it. So I think it's always a good practice to not let agrobacterium sit around. We mentioned about the glycerol stalks and doing your electroporation. So I think the sooner you resuspend them, the better. So I can't say from experience because we always just follow what we were given and are kind of good practices. So yeah. Okay, so
okay, great. Yeah, so we're a little bit over the hour, so I think we'll close out there.
Move to the next section on antibiotics. It's okay. I mean, we can just keep going. It's just that I have it hard stop at 12:30. So we have
sure.
the antibiotic section from Wayne.
Oh, yes. Oh, sorry. Apologies. Yeah.
Okay,
here we
that's
go.
okay. All right. So I'm going to cover this for Wayne. Wayne Parrott put this together. Thank you, Wayne. Hope you're feeling better. And I'm just going to talk about antibiotics. So Wayne has some really good information here that you'll see about different antibiotics, about killing agrobacterium, or maybe not killing it, but definitely holding it back when you do your transformation. And then he lists some antibiotics here to select for engineered cells. I would say that kanamycin in that backbone with that NPT2 gene is the most common for a lot of vectors. Some of these spectinomycin, some are not as common, something like hygromycin, but just know what that selectable marker gene is on the backbone of your plasmid, as that's important to select. There's also there was a question in the chat about, some agrobacterium strains have their own antibiotic resistant markers. And there's a really good paper that Wayne lists down here in the bottom left-hand corner. I have the one page printed out, and it's on my bulletin board to let me know that which different strains have different bacterial markers for the agrobacterium. I can share an interesting story. It's as important because I had a postdoc who asked me to help troubleshoot. He was having this is a postdoc from another lab, and that lab was not set up to do plant transformation. So he was trying to do this following some information that he found. And he told me that he did so many experiments and really large experiments and was getting a lot of agrobacterium overgrowth. So I helped him troubleshoot, and I asked him which strain he was using, and he said he was using AGL1. And I said, all right, we use that a lot. We don't have a real problem with overgrowth. I said, which antibiotic are you putting in the medium to stop the growth of that agrobacterium? And he said, well, I'm using carbenicillin. And I said, there's your problem. AGL1 agrobacterium is naturally resistant to carbenicillin. So by you putting carbenicillin in the medium, it's not really doing anything to keep back your AGL1. So it's really important when you're using an agrobacterium strain to know what that natural resistance would be. One that we use commonly in my lab is Timentin. And Wayne put up here in the right-hand corner what that is a mixture of. But again, we use primarily AGL1, and it works really well for the Timentin does for holding back the growth of AGL1. Amy, next slide, please. Okay. Can you just go through the animations so that they're all there? Right. So again, Wayne listed here showing that there's genomic DNA in the agrobacterium that has got this natural resistance. But basically, the take-home message from this is know your agrobacterium, know the plasmids. Look at those maps, know what you're using for your antibiotic selection. Sometimes people say, oh, well, and knowing what's, because you've got your backbone on your plasmid and what that selectable marker gene is. And then you have a different selectable marker gene in your tDNA for your plant transformation. Sometimes they're both NPT2. Sometimes, the backbone will call for NPT2, but your tDNA might be hygromycin. So really, again, big message, know your agrobacterium, know the plasmids that you're using to make sure you're getting the correct selection. Next slide, Amy. And then Wayne provided a list here of kind of like how they keep track of their strains, their plasmids. This is just an Excel sheet that they're using from his lab. And it's really a good reference because, as it was mentioned before, make these charts not just for you, but for others in the lab. And especially, we know, in labs, we have graduate students who move on, postdocs who move on, technicians, and having to not go through somebody's lab notebook to find out what they did is a real benefit, and having some sort of database. And this is a database, whatever's going to be useful for your lab. We're not making any recommendations. I think everyone finds what works best for them. And this is Wayne showing kind of a simple Excel sheet to do that. Next slide, Amy, please. And then, as Bill mentioned earlier, keep in mind when you're using antibiotics, tetracycline is light sensitive. Be aware of the same brand, using the same brand and the supplier that's recommended. I can give you a story about hygromycin. And using hygromycin, we learned that hygromycin isn't the same from every supplier. Some are more potent, and you have to look at that activity, and some aren't as potent as others. And even this isn't just for bacteria, but even for plant selection, you really need to look at the supplier if you're starting out with a new method, or definitely whatever's in the protocol, follow it. And these are some recommendations that Wayne has listed for stock solutions. And make your stock solution, filter sterilize, freeze aliquots. Again, small aliquots. You don't want to be freeze thawing your antibiotics, similar to you don't want to be freeze thawing your agrobacterium a lot. And you want to add these after autoclaving, not taking the flask out and then adding the antibiotic right to that flask because it's still hot. we use water baths in my lab that cool to a temperature that prevent the medium for gelling. And we basically say, if you can hold it, then it's cool enough to put your antibiotic in, because if you put antibiotic in medium that's too hot, their heat labile is going to inactivate it. And he's given some links down here at the bottom. And again, this is all being recorded and will be available on the PlantGENE website. Before we don't want to rush and end, we have time, a little bit of time for any kind of comments from the panelists that you might want to add to that with the antibiotics in Wayne's section.
Can I add one?
Sure.
And so what was mentioned here to prevent the growth of agro, and so what are the antibiotics to use? And then other way to manage that is to use the auxotrophic strains. So that is very instrumental and have been very, very successful. So you can select the Agrobacterium strains that are auxotrophic, and then you will be able to manage overgrowth. So in order to do that, you have to plan in advance when you are making the plasmid and introducing in the Agrobacterium. So working through that will solve a lot of issues.
Okay. I saw a question here in the chat about, is there a problem when your backbone of your plasmid has the same selectable marker gene as your TI plasmid or your tDNA? Good question. No, and that happens pretty commonly if you're selecting for your bacteria. It's often it's NPT2 on the backbone and then NPT2 in your TI plasmid, so in your tDNA. And I think that's what that question was asking about. Let's take time for one more question, Amy, if you want to choose from the chat, and then we'll wrap this up.
So there was a question here from Sudeshi. How long would plates with antibiotics last in the fridge?
We don't do a lot of storage in the fridge. Does anybody do store? We basically make our medium fresh for preparing, but yep. Does anybody have an experience?
I have used like a solid media with antibiotics prepared a few weeks before the uses, but it's not definitely recommended. Some antibiotics are more fragile, they don't last long, but like a kanamycin, spectromycin, maybe they are stable up to a few weeks.
You learned anything?
Sorry, Nigel was unmuted. Unless Nigel, if you want to add, you can just unmute yourself. But I think he's on the phone.
Okay.
So I had another question in the chat from Jitesh. In your experience, how does exposure to light during agrobacterium processing after growth affect transformation efficiency of plants?
So we always do our, I think he's talking about co-cultivation maybe, after we do the infection. It's at least we do everything in the dark. We're culturing, so I'll just start from the beginning. When we do our electroporations or we're doing, growing our agrobacterium, it's always in the dark on the plates. And then when we do plant transformation, our co-cultivation is done in the dark. I know there are some protocols where people do their co-cultivation in the light. We don't do that. The way I was told when I was first learning about this is agrobacterium evolved in the soil. And it grows more efficiently and effectively in the dark and cooler temperatures like we do 19 degrees C. But I know there are variations out there. I can't speak to them because we haven't tried that. Okay. But in general, all of our agrobacterium, whether our bacterial cultures, whether they be E. coli or agrobacterium, are done in the dark.
Okay. Great. Yeah, thank you for that. And then there was just a comment here, so maybe you can expand on this. So the comment was, you cannot have the same antibiotic resistant genes on both the TI plasmid and the binary plasmid. I think it's because you don't know whether the binary plasmid is actually in the agrobacterium cells.
I did, Joyce did answer that, and I also wrote the message that, if you have created a glycerol stock and then stock and everything, so generally, people do tend to use few antibiotics. More you put, it is causing a stress on the agrobacterium. And then this should become very relevant if your efficiency, you did not get the impact, like expected efficiency. And then utilization of these antibiotics are to ensure there is a presence of binary vector, TI plasmid, and all those things. So it is important knowledge to have when you are troubleshooting. So it should not be ruled out, but it should be kept into consideration.